Okay, the last thing we are going to talk about in Chapter 3, besides just a couple of pathologies, are neurotransmitters and neuromodulators. So, neurotransmitters and neuromodulators are the chemicals that convey information among neurons and at synapses where neurons synapse with other cells. So, a neurotransmitter, it's released by the presynaptic neuron and it acts directly on the postsynaptic membrane, either by opening ion channels or by activating proteins inside the postsynaptic cell. Neuromodulators, on the other hand, are released into the extracellular fluid, so they're not released into the synapse, they're just released into the extracellular fluid, and they run around and adjust the activity of a whole bunch of neurons in the area. Okay, so neurotransmitters, they have a specific target. Bam! They want to get to the receptors on that postsynaptic membrane. Neuromodulators are kind of running around hitting everything that's in the area. So neurotransmitters, they affect the postsynaptic neuron either directly by activating ion channels or indirectly by activating proteins inside the postsynaptic neuron. So there are two different types of neurotransmitters: fast acting, that act directly, and the transmission requires less than a millisecond, which is 1,000th of a second, really fast. And then there's slow acting, the ones that act indirectly, and it's 1/10 of a second to minutes. So they're much slower acting than the fast acting ones. Okay. Neuromodulators, they alter neural function at a distance away from the synaptic cleft. So in the little diagram, which we'll go back to, it shows some neuromodulators, substance P in this case, which we'll talk a little bit more in detail about. Not much detail though. And it is acting away from the synapse. It's influencing that presynaptic terminal in some way, either inhibitory or excitatory, and so the neuromodulator is acting away from the synapse, okay, or away from the synaptic cleft. So the effects are more slowly manifested and they are more longer lasting than those of neurotransmitters. So really the main thing I want you to get is that when you compare neurotransmitters and neuromodulators, is that neurotransmitters happen fast, their effects happen fast, and they don't last as long. Neuromodulators, the effects are a little more slow and they last longer. Okay, that's all I want you to get on the distinction between the two. So talking about specific neurotransmitters and neuromodulators, there are only a couple that I want you to know. Acetylcholine is one of them. It's used at the neuromuscular junction. It's usually excitatory. And the neuromuscular junction almost always fires when you get a nerve impulse to it. A neuron may or may not fire when you get a nerve impulse to it. So acetylcholine is an excitatory neurotransmitter, and it's used at the neuromuscular junction. It's also used throughout the autonomic nervous system, but we're not going to worry about that right now. There are some amino acids, and they're all G ones: glutamate, glycine, and GABA. Glutamate is excitatory, glycine and GABA are inhibitory. We're going to talk a little bit more about glutamate in Chapter 4 and how it functions in strokes and other areas where there's damage in the brain. But other than that, we are not going to talk much about glutamate. But I want you to know that acetylcholine is excitatory and it functions at the neuromuscular junction. So of the slow-acting neurotransmitters, there are the amines, and a lot of these we've heard of: dopamine, serotonin, norepinephrine, histamine. So when we say slow-acting, that's just in relationship to the acetylcholine, and they're the ones that act indirectly by activating the release of proteins inside the cell rather than activating the opening of ion channels. Okay, peptides are hormones, neurotransmitters, neuromodulators. The only ones that I want you to remember are substance P and the opioids, which are our endogenous opiates. Those are things that help control our pain that our brain is producing itself. The rest of those, there's lots of cool information in the book, but you do not need to worry about that. So substance P and the endogenous opioids, that's the only ones you need to know about. There's this Venn diagram in the book, and I don't expect you to memorize this or anything, but I just think it's kind of neat. When you see how serotonin and norepinephrine and dopamine interact to control our emotion, mood, and cognition, and how they function in some different pathologies too. So it is really just kind of neat. Those three neurotransmitters are controlling a lot of our emotion, cognition, and mood. So you don't have to memorize this. You don't have to know this for the test. I just think it's interesting, and that's the only reason I'm showing it to you. So substance P we're going to talk about because it is functioning a lot in pain, and in physical therapy we deal with a lot of people in pain. So it's an interesting thing to know about. It's one of the most common neuropeptides. It stimulates nerve endings at the site of injury and then in the central nervous system. And it acts as a neurotransmitter. So it can be a neuromodulator or a neurotransmitter, as can some of the other ones, carrying information from the spinal cord to the brain. So a lot of our pain control mechanisms, which we will talk way more about in chapters 6, 7, and 8, have to do with whether or not pain information is being carried from the body to the brain, and of course it has to go through the spinal cord. So substance P has been strongly implicated, like they like to say, as a neuromodulator in the pathological response to pain syndromes. So something like fibromyalgia or hypersensitivity syndromes, which involve perceiving normally innocuous stimuli as painful. So something like light touch in a normal nervous system will be perceived as pain, whereas in a pathological nervous system it's perceived as pain. In a normal nervous system it's just perceived as light touch. And we will talk about this way more later, but that's the interesting thing about substance P. It's an identified neuropeptide. It's one that functions a lot in those pathological pain response syndromes. And so it's potentially one that can be targeted by medications. So it's an interesting thing. Synaptic receptors, in the book they really talk a lot about specific receptors and where their sites of action are and that sort of thing. We are not going to go into that, but I just really, all I want you to know about synaptic receptors is that they are typically named for the neurotransmitter or neuromodulator which they bind to, or to which they bind, and they produce either direct or indirect actions. So when they act directly, the receptor and the ion channel are a single functional unit. So when the neurotransmitter or neuromodulator binds to it, it opens an ion channel. That's that fast-acting direct receptor. The ones that act indirectly, they stimulate a cascade of intracellular molecules that either activate ion channels or cause other changes in the postsynaptic neuron. Okay, so this is really the same thing, just as a review. Postsynaptic receptors, they either directly open ion channels, that's fast. They indirectly open ion channels by using that second messenger system, or they activate a cascade of intercellular events, whatever those events might be. Okay, so there are a million little charts and examples and diagrams in chapter 3 that we are not going to go over. The only one I want you to look at, just as a way of showing the different things that could possibly happen from a neurotransmitter receptor, is on page 59 in chapter 3, and it's right here on the screen right now. Um, so the neurotransmitter that binds to a receptor that's an ion channel. It briefly opens the ion channel. It causes local either depolarization or hyperpolarization of the membrane. So that's that quick, direct, fast-acting thing. The G protein mediated receptors, those are the ones that are indirect, and they so they can do a lot of different things that are listed on this chart. I do not want you to waste your time memorizing this chart. I just want you to know that a direct effect, a direct receptor opens an ion channel and either depolarizes or hyperpolarizes the cell membrane. An indirect releases a cascade of intracellular events that can do other things besides open an ion channel. Okay? So it's either open an ion channel or other stuff. That's all I want you to know about it. There are medications that are neurotransmitter agonists and antagonists. So obviously the agonists are drugs that amplify the effect, and antagonists are drugs that decrease the effect of the neurotransmitter. So the agonists a lot of times will mimic the effects of the naturally occurring neurotransmitters. So a really good example of this is the carbidopa and levodopa that goes under the brand name of Synaptic that is used to treat Parkinson's. It mimics the effects of naturally occurring dopamine in the brain, and that is what is missing in Parkinson's. The cells that create dopamine are dying off, and that's what creates your motor effects. Okay, so that's an agonist drug. It's one that mimics the effects of the naturally occurring neurotransmitter. Antagonists, either prevent the release of neurotransmitters, or they bind to the receptor and impede the effects of the neurotransmitter. So a good example of this is the stop smoking drug Chantix is actually a neurotransmitter antagonist. So it binds to the receptors that nicotine usually binds to, and so it doesn't let the nicotine bind. And so you don't have, so when you're on Chantix, you can smoke a cigarette and it's not going to do anything for you, whatever it normally does. The nicotine's not going to bind to its receptors because the Chantix is already bound there. And so it makes you not crave cigarettes because they don't have the effect that they normally have, that the nicotine normally has. And for some reason it also causes you to have weird dreams and stuff, but because it's messing with your neurotransmitter receptors. I mean, that's what it really is. So those are two good examples of a neurotransmitter agonist or antagonist that you see every day on TV or, you know, hear about in the clinic. So kind of interesting. So when you talk about neurotransmitter agonists and antagonists, there are some different diseases that we will talk about. Lambert-Eaton syndrome, it's an autoimmune disorder which destroys the voltage-gated calcium channels in the presynaptic terminal, so it's going to inhibit that release of neurotransmitter. We're not really going to talk much about that one, but myasthenia gravis is another one, and it actually attacks and destroys the acetylcholine receptors at the neuromuscular junction. So there are fewer receptors, you get less of a muscle contraction. It's pretty interesting. There's a little video in the Canvas classroom that talks about myasthenia gravis and a little more information, which we'll go over in a second. The other one that they bring up in the book, and we're not going to talk much about this either, and you will not be tested on this, I just think it's mildly interesting. It's called channelopathy. It's a disease that involves dysfunction of ion channels. Frequently, it's a genetic disorder because those ion channels are proteins that are coded for in the DNA. It's a genetic disorder or genetic mutation, and it can cause some cases of epilepsy and migraine headaches. So pretty interesting in that respect, but you will not need to know anything about that. It's just a point of interest. Okay, so there are some videos about the function of neurotransmitters and some specific neurotransmitters and neuromodulators. Again, anything that's in those supplemental videos that is not covered in the lecture or is not covered in the book or isn't on the objectives, you will not have to know that. It's just additional information. Okay, there's a listing of specific neurotransmitters and neuromodulators and what they do. So I'm going to go through a little bit of them. Acetylcholine, it's used at the neuromuscular junction with all of them. It also acts as a neuromodulator and autonomic function, controlling your heart rate and some other things. In the central nervous system, it functions in the selection of objects for attention. It's usually an excitatory neuron. So when you're getting all this sensory input from the outside world, your central nervous system has to decide what it's going to pay attention to and what it's not. And acetylcholine is part of the pathway on that. Pretty interesting. So dopamine is a neuromodulator in motor activity. It's in motivation and cognition, sort of the pleasure and reward cycle. If you get a chance to look at that mouse party link, you can see that a lot of the drugs that, recreational type drugs or whatever you call them, they affect dopamine. They affect that pleasure and reward system, which is why people get addicted to them, because they want that effect. So loss of the dopamine producing cells in the brain, that's the primary deficit in Parkinson's disease. So it causes involuntary muscle contractions and tremors and that sort of thing. So with schizophrenia, dopamine, there's an excess level of dopamine and it creates psychosis. And that you can also have involuntary muscle contractions and pacing with that as well. And unfortunately, Parkinson's disease, if you're on the medication for a long time, it can start to cause hallucinations and psychoses. So it's that excess of dopamine. So the medications that they use to treat schizophrenia often block dopamine action. So really, it's a wide-ranging thing in our mood and motivation as well as motor activity. So amphetamines increase and decrease the or release of dopamine, and they decrease its reuptake. So it gives you more energy, more motivation. ADHD is often treated by low-dose amphetamines, and that actually increases the release of dopamine and decreases its reuptake. Kind of interesting. Cocaine also decreases the reuptake of dopamine, and dopamine can be excitatory or inhibitory depending on the receptor that it binds to. Okay, so serotonin is a neuromodulator for general arousal level and alertness. Low levels can be associated with depression or suicidal behavior. Prozac and Zoloft and all those ones, the SSRIs, they block the reuptake, and so the serotonin is in the synapse longer. It gives it more time to bind to the receptors. It's usually an inhibitory neuromodulator. So you can see something that affects. One of the things that they always say with those SSRIs is that it might cause suicidal behavior in teens or young adults. This is all. There's stuff going on with that. There's all this stuff is going on in our brain, and serotonin affects it. So with norepinephrine, it causes increased attention to sensory input, contributing to improved surveillance of surroundings. This is one of the big neurotransmitters in our fight or flight reaction, and we will talk about that in the autonomic nervous system chapter. We will talk about the sympathetic nervous system and the fight or flight. Excess of it produces fear and panic, and it can be excitatory or inhibitory depending on the receptor. So that's really important in our survival mechanisms. Substance P, which I talked about a little bit earlier, it functions as a neurotransmitter in the nociceptive pathway from the periphery to the spinal cord and possibly up to the brain. The nociceptive pathway is the one that transmits pain. It's a pain transmitting pathway, and we will talk about that more in chapter 6. As a neuromodulator, substance P is considered to be one of the main contributors to chronic pain syndromes. It's a long duration excitation of postsynaptic membranes, so it's usually unexcitatory. Substance P, it's neuromodulator. It's floating around, affecting all the neurons in the area. So endorphins, those are our endogenous opioids. They inhibit neurons in the central nervous system that are involved in pain perception. They're usually inhibitory. So the synthetic opioids, or the naturally occurring opioids, bind to the same receptors as our endogenous opioids. So when we take, Vicodin or whatever narcotic pain medications that are out there, that's what it's doing. It's binding to the same receptors that our natural endorphins use. So there's a short little video on the different types of neurotransmitters, if you want to review those. The receptors, the main thing I want you to remember about receptors is the action of the neurotransmitter depends on what type of receptor it binds to. That is the main thing. I guarantee you that will be on the test. So an action of a neurotransmitter is determined by the receptor that it binds to. Remember that. Live it. Okay, so we talked about this a little bit earlier. This is we're just reviewing it. Synaptic receptors either directly open ion channels, those are the fast-acting ones, or they activate the G proteins and cause that cascade of intracellular events. Okay, so the ligand-gated ion channels, remember ligand is something that binds, they are a gate. So when something binds to it, the gate opens. When the neurotransmitter gets taken up again by the other cell, the gate closes. Okay? So G protein mediated receptors, there is a ton of detail in the book which you do not need to know. All you need to know is G protein mediated receptors can do a ton of different things in the cell. They can activate, they're highly complex, they can alter a variety of cellular functions. That's all you need to know. Okay, there's a couple little videos about Myasthenia Gravis. We do not have a shortage of acetylcholine in Myasthenia Gravis, but the receptor sites are damaged by the immune system. It's an autoimmune disorder, like a lot of the other neurological deficits. There's a short little 6-minute video kind of on what it's about, how it works. There's an 8-minute video of a neurologist examining a patient with Myasthenia Gravis, and it's pretty interesting. I've worked with 2 people with Myasthenia Gravis in my relatively short career, and it's surprising because it's a fairly rare illness. It's actually treated by immunosuppressants, which preserve those acetylcholine receptor sites. And people respond really well to medication. A lot of times when you're seeing someone in therapy, the 2 people that I've seen in therapy, they were waiting. One of them was an inpatient, one of them was an outpatient. The inpatient was inpatient at a subacute facility. They waiting till they got her medication sorted out so she could go back home. She lived alone. She was an older lady, and one of them was a younger person who lived with a supportive family. I mean, she was in her 50s, but the other lady was in her 80s. And she actually came into PT because she was falling a lot. And her doctor didn't know what to think about it, and the physical therapist was actually able to help determine the cause. And then she got we worked on strength. You want to work on strengthening with myasthenia gravis people, but you don't want to tire them out, because the more times the muscle contracts, the fewer receptors there are available and the weaker the muscle gets. So a lot of our postural muscles and things like our eye muscles and our swallowing muscles get tired out fast because we use them a lot. And so you want to work with people with myasthenia gravis working on energy conservation. A lot of times working with them earlier in the day is helpful because they haven't worn out all their receptor sites yet, basically. And you're working on modifying their activities until their medication gets sorted out. So it's really interesting and very unusual that you it's very unusual that I've gotten to work with 2 people with myasthenia gravis. So we are I talked a little bit about the homework assignment. I'll let you read through it, and hopefully it'll be kind of a fun assignment because you get to be creative with it and use your own words to describe it. And students in the past have done very creative analogies, made up stories with characters, done all sorts of things. Nobody's ever done an interpretive dance, but that would be a little crazy, wouldn't it? But hey, go nuts if you want to do that, or use sock puppets or Lego figures or whatever you want to do. Whatever it takes for you to explain those events in your own words, and by doing that, hopefully it will help you understand the process. So that's it for Chapter 3. There's a quiz for Chapter 2 and 3 in this module, and then we will move on to Chapter 4, which is neuroplasticity. Exciting stuff.